Welcome everyone, it's me Kim Ali from Tuskegee University, and today I'm going to present an article by Gungliandolo et al. 2021 on the topic of effect of cannabidiol CBD on canine inflammatory response and ex vivo study on the LPS lipopolysaccharides stimulated whole blood. And this presentation is done for the graduate seminar second course at Tuskegee University. So the CBD is one of the major non-psychoactive plant-derived cannabinoids, and it is known to act as anti-inflammatory and immunomodulant agents. Thus, it can affect both innate and adaptive immune response. The immune-modulating, anti-inflammatory, and other biological activities of CBD has been characterized most often in the human medicine, but there are very few research in veterinary medicines. The pharmacokinetics and tolerability of CBD has been characterized in the dogs, but there are only few studies on the therapeutic use of the CBD in dogs. So, the objective of this study was to evaluate the effect of CBD directly in the canine inflammatory response. To achieve this objective, the researcher did the ex vivo whole blood culture with the stimulation by LPS, focusing on the level of major regulating cytokines, the two pro-inflammatory cytokines, that is IL-6 and TNF-alpha, and the anti-inflammatory cytokines IL-10. For this, whole blood was collected from 6 healthy dogs, which are of German Shepherd breed, and they are 2 to 10 years old, and 3 were male and 3 were female. Thus, the collected whole blood was cultured with or without stimulations, and in first control group the whole blood was cultured only with the vehicle. In the second group, the whole blood was cultured with LPS stimulation and vehicle, and in third group the whole blood was cultured with LPS of 100 nanogram per mil and along with the treatment of CBD of 50 microgram per mil. In the fourth group, the whole blood was cultured with LPS and with the treatment of CBD of 100 microgram per mil. The different level of IL-6, TNF-alpha and IL-10 was measured by using the sandwich ELISA kit from the Quantikine R&D system company. And here is the result and discussion part. In the first graph, we can see the level of IL-6 on the different group. We can see that when the blood culture is stimulated with LPS, the level of IL-6 increases significantly, but with the treatment of CBD, the level of IL-6 goes on decreasing significantly. In the second picture, we can see the level of TNF-alpha. Here also, we can see that the TNF-alpha increased significantly when stimulated with LPS and is not treated with CBD, but with the treatment of CBD, the TNF-alpha level decreased significantly. As we know that IL-6 and TNF-alpha play the key role in physiological inflammatory response, such as during the infection, and are also associated with the poor clinical outcome. And the results showed the significant lower level of secretion of the IL-6 and TNF-alpha when treated with CBD, which is most desirable. In the first graph, we can see the level of IL-10. Here, when stimulated with the LPS, we can see the significantly increase in IL-10 level, but with the CBD treatment, the IL-10 level increases but are not significant as the second group. In the second graph, we can see the TNF-alpha to IL-10 ratio. The ratio increased significantly when stimulated with LPS, but it goes on decreasing when treated with the CBD. The level of IL-10, which is an anti-inflammatory and immune-regulating cytokine, was not significantly altered by the CBD treatment. And the balance between TNF-alpha to IL-10 ratio is very important for maintaining the immune homeostasis. Thus, the result shows that LPS stimulation can produce significant increase in TNF-alpha to IL-10 ratio, while the treatment with CBD is able to significantly reduce the ratio, which is desirable. Here are the limitations of the study. There are no accurate study on the cytotoxic effect of the CBD if present, and there is limited use of CBD concentration, limited sample size and species differentiation, and also the sources and preparation of CBD may vary. The conclusion of this research is CBD was able to reduce the inflammatory response induced by the LPS in canine whole blood. Thank you.